Good morning, I'm Rishita Agarwal and I'm roll number 16. I'll be showing you my project on the Iris-Dataset Dashboard. So my code is in R and I have used Shiny to deploy the dashboard as well. So let me start with the processing first. So I've processed the Iris dataset in such a way so that I can store some of its data in which I can show in the interactive dashboard. So initially the iris, I've added few more categories here, like ID. I've used species to factor those and factor the classes, and also I have created sepal area and petal area columns. The next thing that I have done is iris summary. Here I have grouped by on the basis of the species and calculated the mean, standard deviation, minimum and maximum of all the columns which are numeric. And I've saved these new created datasets into QS files. So QS files are basically very compressed files which help in memory management as well as for faster loading of the data. And I have also stored the metadata here. So as you can see, I'll run this file now. So initially this is my data directory where the iris and the metadata are empty. So now I'll run this file to show you that the iris and metadata are updated as I run this file. So I'm running this file and you can see that now there is iris processed, iris raw and iris summary. In the iris directory and in the metadata directory I have meta.rds. Now, since these files are formed, I'll now go to the another processing file that I've made for Iris Analytics. This file is specifically for the data in the analytics section, which I'll show you on the dashboard in a while. So here I have again used Iris processed, which I have loaded from the QS file that I've stored, and I've mutated a few more columns here, like the z-scores for the length and the width of all the sepals and the petals, as well as the outliers, also the ratios for the area, length and width. And I've again created a QS file to store it. Now I'll show you that this analytics file is right now not present in the iris directory. I will run this and show you that it is formed right now. We can see the analytics file is formed. So these are QS files which are now completed, and we'll now see how the dashboard looks like. So I'll initially run a local version here and then later also show you the deployed website. So in the run.r, I have created a bash kind of a script where I'll run this particular R script and it will automatically run my RMD file. So let me also give you a show of my RMD file. So this is my Iris-Dashboard.rmd file. Here I have used the Shiny library as well as the Flex dashboard to show better view of the dashboard. Yes, so these are some of my libraries which were required in this entire graph on the dashboard. These are my data which I have processed here, which I have loaded here. So this is the entire flexboard kind of a structure where I have created a sidebar for all this inputs, for taking the inputs, and also I have scatter plot, I have a distribution plot, a correlation plot, and box plots along with the summary statistics. So so let me show you the dashboard first in order to tell you more about these plots now. I'll run this run.r file now. As we can see, the local host has started running and we can see the dashboard in a bit. Yes. So this is the dashboard which I have created. Here, firstly, initially to tell you about this iris dataset, I have written some basic things about the iris dataset, that it contains 150 observations. It is a small dataset having 4 features, which are sepal length, sepal width, petal length and petal width on 3 different species of flowers, which are Setosa, versicolor and virginica. This dataset was created by biologist and statistician Ronald D. Fisher and he published this in the 1936 paper, The Use of Multiple Measurements in Taxonomic Problems. So now let's see all these interactive scatter plots and distribution plots. Yeah. So let me show you the deployed website as well. This is the local version. I'll show you the deployed website for a better view of all these plots. So this is the deploy.r file where I have actually which I have where I have actually deployed the project on Shiny apps. So this is the link of my deployed website. I'll go here and show you further. So let me now show you the deployed website dashboard, and this is my deployed link of the dashboard, and it's loading. We can see that this is again the one which I showed you on the local, and here you can select the species. You can change the x and y variables on the basis of what what comparisons or what data you want to see. So I'll go on with this one by one in each plot. So starting with the sepal, let me start with setosa and virginica. We can see sepal length and let's see petal length. I want to compare between the sepal length and the petal length. I can see that this particular z-score is the normalized score here. We can see the species is virginica, the blue one, and the green one is setosa. We can see all the data points and a green trend line here. The trend line tries to show you how much one of the feature in the y variable affects the feature in the x variable or the vice versa. So we are seeing that how much the petal length affects the sepal length. So we can see here in the distribution plot that the sepal length for the virginica is overall much more than that of setosa. That means we can come to a conclusion that virginica usually has a sepal length about here, like let's say 0.56. The sepal length is more, but the density of sepal length in setosa is concentrated at a particular point. Let's say that is approx a sepal length of 5 is more prominent there. Here the distribution is more or less kind of similar on a similar scale. And in the box plot, it is very clearly visible that there's this outlier, and the virginica median is clearly very much above the setosa median in the box plot. So this is the correlation matrix, and let me scroll it down to see that we can see that the blue ones indicate a very high correlation, whereas the green ones indicate a very negative correlation. So here petal length and sepal width, if we'll see, it shows a correlation of minus 0.49. That means it has a let's say a negative of zero point 0.5 approximately correlation, which is quite a inverse relation they have. On scrolling this down, we can very clearly see that sepal area is more or less independent of other variables like petal length, petal width, which is quite obvious because sepal area is made up of sepal length and sepal width, so it's more focused on those areas which we can see clearly that sepal width and sepal length are a bit more prominent in comparison to other correlations with petal width or petal length. We can, when we scroll, we can see the sepal width to be very negative correlated with other factors like petal length, petal width, or petal area. So why is this observed with sepal width is one of the things that we can conclude after seeing this is that the sepal width is kind of inverse to all these things. Like if the petal length is more, then the sepal width will be less, or the petal width is more, then the sepal width will be less. So in that way, we can conclude this thing. Another observation which we can see is that that for all the three species, the summary statistics are kind of constant. For all the three species, we can see the setosa has a sepal length mean of 5, virginica has 6, like it's an increasing order in this particular form. But when we come to the sepal width, we'll see the sepal width of setosa is much more than those of virginica or versicolor. So this is something that we can say about the sepal width of setosa, that it is in general very high. We can also see that from the graph. Let's change the x variable from sepal length to sepal width and see that the graph where in the sepal length graph the setosa was much less than virginica, But here, let's also include versicolor and see the comparison between these three. So setosa is clearly has more sepal width than, as compared to versicolor and virginica, which was not the case when the x variable was sepal length, which is a kind of an approximately inverse relation here. So this is what it is. And talking about the analysis summary, so here the statistic and the p-value kind of tells you that these factors are very uncorrelated, or sorry, very correlated as in that they affect each other, but as species they are very uncorrelated. So the correlation coefficient here shows that the features themselves, let's say sepal length, sepal width, petal length, and petal width themselves are very connected to each other. That means we can come to a conclusion that they are not exactly uh, randomly sampled or anything. The data set is very good and it reflects that how much these factors affect each other in that form. We can see the outliers are very less in number, which again indicates that the data set is balanced, and yes, that's what it shows. Here on seeing the trend line, let's see the trend line for a single uh, species. For let's say setosa, we can see the petal length and sepal length here. The trend line shows a R square of 0.071, which is pretty less, and also you can see the scatter plot is pretty much all scattered around, not in a particular direction. So which shows that the correlation of petal length and sepal length would be very less. Let's also cross check that from the correlation matrix. So petal length and the sepal length, let's go there. Yes, we can see it's only 0.27, which is fine, but like we can kind of conclude that there is less correlation between these two things. We can also check this for some other things, like let's go from petal length and sepal width. Sorry, sepal length and sepal width. So here we can see that the trend line is going towards a particular distribution of the data, and the data distributed is kind of pointing towards a single direction. We can also zoom out and see this more clearly. Yeah. So a zoomed in version is this. And we can also cross verify sepal length and sepal width correlations here. So let's go sepal length and sepal width. We can see 0.74, which is much more significant than that of 0.27, which was between petal length and sepal width. Here we can see sepal width and sepal length are much more correlated. So this was for setosa. Let's check for setosa and versicolor both. And here between the sepal length and sepal width, we can again see here's versicolor and here's setosa. So both of them are in a particular single direction and not overlapping. So we can also say that these the features which are not overlapping for two different species are kind of distinctive features, are the features which can decide whether these two how these two species can be distinguished between each other. We can see from the box plot also that they are very different from each other and do not co-overlap each other. So we can also check the same thing for petal length and petal width. We can see this shows very clearly a high correlation and a high direction. We can clearly see that the petal width for setosa is so less than as compared to versicolor, and both of them are highly correlated, the petal length and the petal width. We can also check from here petal length and petal width. Yes, 0.98, that's really high correlation, approximately 1 ka correlation there is, and the trend line is 0.959, which shows that the petal width affects the petal length up to this percentage, which is pretty high. And the distribution also shows that this petal width can be a very significant factor to distinguish between two different species like setosa and versicolor. Uh, yes, so we can also include virginica here and see that the differences between all these three can be are very visible in petal width, although there is some overlap here in virginica and versicolor in this particular area. So let's compare virginica and versicolor here for some other factors which can help us identify a distinguishing feature. So sepal width is clearly too much overlapping and cannot be used as a distinguishing feature here. The same goes for sepal length. The petal length also. So I think the petal width was the most, let's say, most best option here to distinguish between versicolor and virginica, because all other factors are clearly too overlapped. So, seeing all this, I think it is clear that this interactive plot can highly help us in figuring out how correlated the species are, or the features are, or how much a species can, on what factors the species can be distinguished from each other, and what the outliers are. So basically, this whole data dashboard also shows us that how good of a data set this Iris data set is, how balanced, and how much like less number of outliers display a more unbiased kind of distribution in this particular data set, which shows how nicely this data set has been formed. And that is the reason it is such an important data set in machine learning models, that it is used very frequently for data distribution and data analysis. So I hope I was able to show you my display of my understanding of this particular data set and these dashboards. Thank you.